Okay, hello everyone, and today my topic is uh, oil, oil spills, the invisible impact on the base of tropical marine food webs. And at first of all, I want to say this paper is not talking about a big area of oil spills. What we care about is this tiny oil droplets, and it has a small diameter, about 200 micrometers. And most important is it is very hard to clean up and it is invisible to human eyes. Okay, now the purpose of this study is we want to know how the oil spills affect zooplankton and further influence the marine food webs. Sampling. The sampling location is at Brazil, east coastline of Brazil, and you can see the green spot over here. We have three locations. The first one is reefs, and the second one is Bay Area, and the third one is estuarine plume. And there are three sampling stations in each location. And the red spot means there is oil site over there. Now, after the scientists collected the samples, they took some pictures. So the panel A and B, we can see there are oil droplets between the zooplankton. And the panel C, we can see an oiled crab larvae with an oil stain in his mouth. Results. So the first one, we can look at the chart of zooplankton abundance. The x-axis shows the three different sampling locations, and the y-axis shows the zooplankton abundance. We can easily see the estuarine area has the highest average abundance. Now, this is the composition of zooplankton. We first look at the legend. There is so many species up there, but the 6 species over there, we can summarize it as copepods because they are under the same order. So now we can say we have 94.2% of copepods in estuarine plume and 96.5% of copepod in Bay Area. And now we have 28.3% of coral larvae in reef area. So let me make a short introduction about what is copepod and what is coral. We are going to discuss these two species in the following presentation. So on the left hand side is the copepod image, and on the right hand side is an image of reef building coral. So this chart is oil droplet abundance. Again, the x-axis shows the locations, and the y-axis shows the oil abundance. We can see in the estuarine plume, the standard deviation is very wide, from 12 to 180. But we can still say that the estuarine area has the highest average abundance. Now, this is the ratio of meroplankton and holoplankton abundance. What is meroplankton? It means larvae, which means a baby stage of some creatures. And what is holoplankton? It means a creature spends its whole life as a plankton. So now we can see the reef area has the highest ratio, about 1.5. And the reason why is because it is contributed by coral larvae. Now, this pictures shows the evidence of zooplankton being affected by oil. The scientists measure the value and concentration of oil droplets is about 315 microcubic meter. So we can see the arrow point to the black dot over there, and this black dot are the oil. Discussion. Recall that we have the high value in estuarine plume, both the oil abundance and the zooplankton abundance. So I'm going to explain why this data is different from other locations. First, we have an enlarged picture of estuarine plume. So we can easily see the 3 sampling station is right at the river mouth. Now we know the estuary area usually has a high nutrient concentration because there are so many substances coming from land, so which why it makes a high abundance of zooplanktons. And secondly, we have an oil site is nearby the river, which is the main reason we have the highest oil abundance in estuarine plume. Now, we know there are 94% of copepods in estuarine plume, so it plays an important role in this area. The copepod will ingest the oil droplets and make the oil be part of organic matter. And this kind of oiled organic matter will more likely to attach to the oil droplets and form oily sediments. And the sediment will go down to the bottom of the ocean. So the oil droplets will form a sediment and go down to the bottom of ocean will affect the benthic creature. And the oily organic matter we just mentioned will also settle to the bottom of ocean, and both of them might cause a possibility that the oil will affect the benthic environment. So we also have some benthic creatures that spend a stage of time as a plankton. For example, we have a crabs that is one of benthic creatures, and we know is crab larvae is a plankton. Now we have evidence. The picture here, a crab larvae is eats oil. So we can make a conclusion that the oil does affect the benthic animal. Okay, remember we have a reefs area in our sampling location. So the reef building corals are benthic animals. So we can remind that we have data showing there is a coral spawning event happening right there. So this is a ratio of 1.5 means we have coral larvae, which may cause as a result that this coral may also have been affected by oil pollution. Okay, here is the conclusion. We know this topic is going to talk about the impact of droplets on the marine food webs. So now we know the zooplankton and most of it is copepods will be affected, and also the benthic animals will be affected, and including the crabs and coral. Okay, that's all. Thank you, everybody.
I see you add a lot of labels and arrows to your chart and making the explanation more detailed and completely. And you use many images to introduce the oil droplet and the creatures, and that's quite good. So overall, I think the speech is smoother than last time.
I think your stage performance is much better than last time, but the time control is less than 10 minutes.